Hello, everyone. My name is Tom Callis, and I'm a clinical science liaison here at Invitae. On behalf of everyone at Invitae and the SADS Foundation, with whom today's program was collaboratively organized, I would like to welcome you to a two-part webinar on new approaches to variants of uncertain significance. Our first speaker will be Dr. Dan Roden, Professor of Medicine, Pharmacology, and Biomedical Informatics, and Senior Vice President for Personalized Medicine at Vanderbilt University Medical Center. His research program studies how genetic variation affects human disease susceptibility with a focus on pharmacogenetics and on genetic determinants of abnormal heart rhythms, especially those induced by drugs. Our second speaker would be Dr. Deanna Lee McKnight. She's a board certified in clinical molecular genetics and the American Board of Medical Genetics and Genomics, and is the Medical Affairs Director for Emerging Clinical Omics here at Invitae. While you'll hear two separate talks today, both presenters will discuss several new approaches to variants of uncertain significance as it relates to cardiovascular genetic testing. But first, just a few housekeeping items. All participants are muted. You can submit questions at any point along the way during today's two presentations using the questions pane of your webinar control panel. We'll do our best to answer as many questions as possible during the single Q&A session that will occur after both talks are complete. Today's webinar will be recorded and emailed to all registrants within 24 hours. It'll also be available on both the Invitae and the SADS Foundation websites. So I know everyone's tired of hearing me right now, so I'll go ahead and turn the slides over to our first presenter, Dr. Roden. We're really excited to hear about your work.
You have to give me the slides.
I'm working on it. There we go. Let's see.
You got it? Okay. Well, thank you very much for the opportunity to talk about something that is actually pretty near and dear to the efforts of our lab in the last couple of years. And as I start, let me just make sure that you know that I'm just the mouthpiece for the work that Andrew Glazer and Brett Crockey have led in our group. And you'll see that I'll quote sort of 5 or 6 papers that they produced in the last couple of years, including 4 this year. These are my disclosures. There is nothing exciting there at all. So this is the problem we're here to talk about, the variant of uncertain significance. This is a genetic test result in a patient with a non-ischemic dilated cardiomyopathy, and they have a VUS in SCN5A and LMNA, so 2 VUSs. This is a gene, SCN5A, that we've spent a lot of time on and we'll come back to over and over again. Loss of function variants cause the Brugada syndrome, gain of function variants or loss of fast inactivation variants cause type 3 long QT syndrome, and there are associations with multiple other cardiovascular phenotypes as well. So the American College of Medical Genetics and Genomics has a classification scheme that allows us to evaluate various kinds of data and various kinds of evidence to assign something as benign or pathogenic or somewhere in between. And you can see the kinds of criteria that the ACMG uses, all the way from population data to allelic data to segregation data, functional data. There's an entry in each square of this large grid, and if I read them all to you, I'd run out of time. So I'm not going to do that. These are data that are widely available and support the idea that multiple kinds of evidence can be used to assign pathogenicity. So the VUS problem has, to put it in context, NOMAD, a large data set that lists variant frequencies in over 200,000 exomes or whole genomes, and is publicly available. Unfortunately, NOMAD doesn't include any phenotypic data right now. So these are data of variant frequencies just in the absence of phenotypes. At the time Leo Starita did this study in 2017, there were 4.6 million missense SNPs in NOMAD, of which 100,000 had an annotation in ClinVar, which is the NIH-supported clinical annotation and reporting website. Of the 100,000 that were listed in ClinVar as having an assertion, only over half of those were VUSs as well. So you can see that the vast majority of SNPs that come along fall into the variant of uncertain significance category. So how do you solve this problem? Well, there have been in silico methods developed. You can ask the question, do variant carriers have a phenotype? And that can be done in families with segregation analysis or across populations using larger and larger sets derived from electronic health records or from biobanks, or in vitro studies, functional studies, looking at one variant at a time or many variants at a time or all possible variants. And in this talk, I'm going to discuss a little bit about EHRs, a little bit about many variants at a time. Then I'm going to end the talk by talking a little bit about what I call the shades of gray problem. So I'm going to start by describing the eMERGE-Seq project. The eMERGE-Seq project is a project of the Electronic Medical Records and Genomics Network, eMERGE. We sequenced 109 Mendelian disease genes and 25,000 participants, 2,500 participants per site at 10 sites. And we return pathogenic or likely pathogenic variants to participants, their primary care physicians, and their electronic health records. We use the electronic health records to identify people who previously did not carry a genetic diagnosis, but clearly should have, to estimate penetrance and to reclassify VUSs, and I'll touch on those in the next couple of slides. This is the overview of the returnable results in the eMERGE-Seq project. So these are the pathogenic and likely pathogenic variants, mostly in cancer or in cardiovascular disease. Cardiac is here, is cardiomyopathies and arrhythmias, and aortopathies largely, and then familial hypercholesterolemia, cardiovascular disease occupies a space of its own. 5 to 6% of patients had a returnable variant, and these are the results at Vanderbilt, very similar to the results across the board, most in cancer and in cardiovascular disease, 6% returnable. We had 10 subjects who unfortunately had 2 pathogenic or likely pathogenic variants. And I'm showing this slide because I want to talk a little bit about FBN1. So FBN1 is the disease gene for Marfan syndrome, and I'm going to talk a little bit about an effort that we've made to try to understand pathogenicity of FBN1 VUSs using the eMERGE-Seq data set. So this starts with the Mendelian Inheritance in Man, gigantic textbook issued annually from Johns Hopkins until it went online, and it's now the online Mendelian Inheritance in Man resource, OMIM. And OMIM lists the symptoms for Marfan syndrome, the signs and symptoms that range across eye, cardiovascular, skeletal, and other systems. So Lisa Bastarash, data scientist in our group, had the idea of creating what she calls a phenotype risk score for Marfan syndrome. So here are 3 patients, one of whom has 4 symptoms that remind you of Marfan syndrome. Another has an aortic aneurysm, which is one of the hallmarks of Marfan syndrome. Another one has no Marfan symptoms. So what Lisa has done is assign weights to each of these symptoms. And the weights she's assigned are derived from our electronic medical record and derived from how common these weights are, these symptoms are in patients with or without Marfan syndrome. So then she does a pretty, what I think is a pretty simple mathematical exercise, and she just creates a score. And the score is, if you have the symptom, then you get the weight. If you don't have the symptom, you don't get the weight. So this person has a total score of 9.6, this person 2.2, this person 0. And when we looked at in a set of cases with Marfan syndrome and a set of controls most of whom we think do not have Marfan syndrome, there's a tremendous separation with a p-value that is 10 to the minus 100th or smaller. And we've done this with a number of diseases, and that work was reported a couple of years ago in Science. So what Lisa has done is take the eMERGE-Seq data set, and at the time she did this analysis, we were about halfway through the data, and there were 14 variants whose pathogenicity was thought to be well understood. 5 that were called pathogenic, and they all occurred in individuals with high phenotype risk scores, and then 9 who had variants that were thought to be benign, and they occurred in individuals with very low phenotype risk scores. So that validates the approach again. And now these are the other 82 variants of uncertain significance across those 15,000 subjects. And you can see pretty clearly there are 2 that really do look like they're pathogenic. So this is the idea of using the electronic health record as a functional readout for deciding whether a VUS is or is not pathogenic. And so it's a promising initial result. To come back to the results at Vanderbilt for a second, we focused a lot of our attention on arrhythmia genes. There are 8 arrhythmia genes in this set of 109 genes that we sequenced. We focused most of our work so far on the 3 long QT syndrome disease genes here, but we've looked at these others as well. We looked across the electronic health records and asked the question, did people carry any arrhythmia diagnosis at all? Not only long QT syndrome, but sudden cardiac death, ventricular tachycardia, high-grade AV block, sick sinus syndrome, atrial fibrillation or flutter, bundle branch block, premature ventricular contractions. Across the set of patients who carry no variants, these are the distributions of these diagnoses in the electronic health record. Across the set of patients who carry pathogenic or likely pathogenic variants, these are the distributions. You can see pretty clearly that KCNQ1 and KCNH2 have an overabundance of these diagnoses, primarily in the long QT syndrome space. About half of these patients carried the diagnosis of long QT syndrome when they started the study, and about half of them acquired the diagnosis in the course of the study or after being returned these results. The other thing that's interesting, I find, is that there's a strong association, or a larger than expected association between occurrence of PVCs and SCN5A variants. That's the P or LP variants. Here are the VUSs. And what's interesting here, of course, is that there are far more VUSs than there are pathogenic variants. For the sodium channel gene, for example, 334 VUSs and 27 P or LP variants. And you can see that there's a slight overabundance of diagnoses for some of these genes compared to the controls here with no variants, but it's pretty modest right now. But the question then is not across KCNQ1 as an entirely, but what about individual variants? So we've gone ahead and looked at individual variants, and here's a list of many variants. Most of them are VUSs. Some of them are known to be pathogenic or not. Some of them have conflicting data. So you can see the G635R, for example, designated a VUS in KCNQ1, almost certainly is pathogenic based on this analysis here. Same with I94V in SCN5A. So again, using the electronic health record to designate variants as pathogenic or not. So let me now turn to some functional genomics that we've been doing, many variants at a time or all possible variants. This is a study that Andrew Glaser has in press in the American Journal of Human Genetics. He took 83 variants in the cardiac sodium channel and asked the question using high throughput patch clamping on a device called the SynchroPatch 384. It has 384 wells, and 10 of the variants that he studied, and you can, using this 384 well format, you can study these variants and replicate in multiple replicates. 10 of these variants we thought were likely to be benign, and they are benign based on peak current density recordings compared to wild type, which is up here and set to 100%. So these 10 validate the system for looking at benign variants, and here's the other variants that he looked at, and they range in from normal function to variants that have approximately half normal function to a large group of variants that are loss of function variants completely. One of the interesting analyses he did, I think, is to compare the frequency of these variants in the literature and in NOMAD. So if you have something that is reported in NOMAD more than 6 times and has never been reported or has been reported only once as a Brugada syndrome case, then the likelihood is that that's a benign variant that has normal wild type function. On the other hand, if there are lots of cases in the literature of association with Brugada syndrome and few cases in NOMAD, then the likelihood is that those variants are pathogenic. And this puts some numbers around what many of us would have expected. And in the end, he was able to reclassify 60 VUSs and leave only 10 of them as VUSs at the end of the study. And what was interesting, we then worked with some structural biologists in our group, and we came up with structural explanations of why some of these variants look to be pathogenic. So this is one really cute example. This is looking down the bore of the sodium channel. This hole that you see is the permeation pore through which sodium ions flow. And if you substitute at this E901 position for a lysine K, you can see that the pore is disrupted. So that's a loss of function variant. There were 4 variants that were insertion of prolines. That means they disrupt alpha helices, and they were all loss of function. And here's another example. The structural biologists tell us that there is a disulfide bridge between cysteine 280 and cysteine 335, and inserting an arginine instead of a cysteine at position 335 means you disrupt that disulfide bridge, and that is also a loss of function variant. So that's a very cool structural, high throughput functional biology story that you'll be able to read about later this year. This is another story. This is a all possible variant story. So the idea here is to use deep mutational scanning of an approach that allows you to take a plasmid and make every single possible single nucleotide variant in that region of in a region of the plasmid. So this is SCN5A, our gene. We made every single variant in a particular region, which I'll tell you about in a second. We then generate a very, very large pool of transfected cells, millions and millions of cells, each one of which carries one of these variants. We've engineered it that way. And then we expose those cells to a drug challenge, which is, I don't have time to talk about. And the drug challenge kills every cell that expresses functional channels at the cell surface. So here, this cell line expresses channels, but they don't get to the cell surface. So that's a loss of function cell, and the same here, but these others are normal functioning, at least for this assay. We did, and then you do next generation sequencing prior to and after the drug challenge and figure out which ones are loss of function and which ones are not. We did this experiment across 12 amino acids in the voltage sensor of domain 4. That's 240 non-synonymous SNPs. 12 of them are stop codons, and then there's another 12 that are synonymous because you take the amino acid that was there and you substitute the same amino acid. So this was reported earlier this year in Circulation Genetics. So what we do is we take the 12 nonsense variants, which we know are loss of function, and we assign them a score in this assay of 0. And how that was done, you have to read the paper. And then we take the 12 synonymous variants, the ones that don't actually change an amino acid, and we assign them a score of 100. And then we take the other 228 variants that are in this experiment, and we assign them scores based on their next generation sequencing data that is done in 6 replicates. So we have a group of channels that we think are gain of function, a group that we assign loss of function, and a group that we assign relatively normal function to. What we then did was we selected a couple of examples from the extremes and did traditional patch clamping. So here is a gain of function variant that indeed turns out to be a gain of function variant, a loss of function variant that turns out to be a loss of function variant, and a total loss of function variant, V1624E, that turns out to be a total loss of function variant. So that validated that approach. And Brett and Andrew have now a paper in press in heart rhythm that's taking the same approach with KCNH2, the potassium channel that underlies the IKR, the herd current. So I want to close by talking a little bit about the shades of gray problem. Clinicians want to be told, is this pathogenic or is it not? And even if we tell them it's pathogenic, then clinicians want to know, well, will my patient get the disease or not? And that's the problem of variable penetrance. So you can assign something as pathogenic or benign, but that doesn't tell you what the patient's going to get because you have to factor in penetrance as well. So Brett Kroenke has been working, well, let me just give you a little bit of background. In a paper that Brett and Andrew produced 2 years ago, they looked across all 1,700 reported non-synonymous variants in the cardiac sodium channel. And here are 4 that are well recognized in the literature. H558R is a variant that occurs in about 25% across all ancestries. Very few reports in the literature of loss of function or gain of function. Delta KPQ, the original long QT syndrome mutation that was described in 1995, very few people have that and are unaffected. 0, in fact, so far. And everybody who has it has a gain of function variant. E1784K, a variant that we actually described first at Vanderbilt, but has been recognized probably around the world as the commonest pathogenic variant in SCN5A. Some people are unaffected. Most people have the long QT syndrome phenotype. Some of the people have a loss of function phenotype as well. And then there's R878C, which has a few unaffected people in the literature, but most people are a loss of function. So this is a Brugada syndrome variant. So that this kind of population data plus functional data allows you to assess pathogenicity and to some degree penetrance. But the penetrance part is hard to establish when you have variants that look like this. Even if they're pathogenic in silico, when you have 1 patient reported in the literature or 2 patients reported in the literature, it's very difficult to get at that. So what Brett has been doing is trying to use basically a machine learning approach to do the following experiment. Take a variant, incorporate the functional data, the structural data, and in silico tools, then make a prior estimate of penetrance. And this is a Bayesian approach that he's developed with the Department of Biostatistics at Vanderbilt. Now, if there are patients, if there are variants that are present in multiple individuals, like the top ones that I showed you on the previous slide, then you can develop a take the prior estimate and develop a posterior estimate. That is an improved estimate of penetrance, and not only an estimate of penetrance, but the likelihood of penetrance. And I'll show you what I mean by that on the next slide. And then what you can do is you can use machine learning approaches that will allow you to take these kinds of data, even in the absence of multiple reports, and get an improved estimate of penetrance. It'll never be 0 or 1 though. So here are 3 variants: L1308F, wild type function, not likely to be pathogenic, in silico, no predictions. I1660V, thought to be a VUS, loss of function variants, predicted to be pathogenic, near some mildly penetrant residues, and then R878C, I already told you, is likely to be a pathogenic variant. So what Brett can do is generate a prior set of estimates using these kinds of data. And the estimates of penetrance aren't a point estimate, aren't like 0.8, but it's a distribution, and the maximum likelihood for penetrance for this one is 0.8, for this one is 0.4, and for this one is probably around 1 or 2%, even for this one. Then you go into the literature, and here there are 134 cases reported. 1 of them is a loss of function. Here there's 17 reported, 5 are loss of function. Here there are 36 reported, and 32 are loss of function. So you can then use these data, the population data, and combine them to an improved estimate of penetrance. And again, the estimates there's still some variability in your estimate. So what Brett's idea is to reimagine the genetic variant annotation variants of a certain significance. So instead of this one being benign, this one being a VUS, and this one being pathogenic, we'll start to think about things in this way. We'll start to think about things as probably benign, probably pathogenic, and then this one here has risk, and then there's uncertainty in the risk, and the uncertainty is captured by this distribution function here. So that is a whirlwind tour through some new tools that we and many others are now using to start to assign pathogenicity and penetrance estimates to variants of uncertain significance. It starts with families, and it will end with families, and it'll end with individual patients. We use computational approaches, we use functional approaches, and we use biobanks. And my hope is that we'll be able to use all of these tools together to improve the care of patients who come to us with VUSs or with other variants whose pathogenicity and penetrance are open for questions. So I'm happy to stop here, and I'll take questions later, I guess.
Dr. Roden, thank you so much. That's super, super exciting, and I'm excited to get to the Q&A session. But first, we're going to hear from Dr. McKnight. And Dr. McKnight, I'm going to turn over the presentation control to you as soon as I can get to it. And while I'm doing that, I'll remind our audience that you can enter in questions anytime for the Q&A session by putting them into the chat box, or sorry, the questions pane, excuse me. All right, Dr. McKnight, there you go. Thank you.
There we go. And you can see me and you can see my screen?
Perfect.
Great.
Yes,
okay, perfect. Well, thank you. Dr. Roden definitely did a lot of my heavy lifting, so I might be able to fly through some of my slides here. So today I'm going to discuss how we at Invitae are reducing variants of uncertain significance through functional modeling, and how this impacts genetic testing for cardiovascular disease. First, I'm just briefly going to talk about variant interpretation and how this happens at a clinical diagnostic laboratory. In general, diagnostic laboratories use the guidelines from ACMG for variant interpretation. These guidelines came out in 2015, and mainly they were an attempt to standardize the terminology that was being used in the industry, but also to provide some recommendations on how to do variant interpretation for different diseases. We at Invitae use Sherlock, which takes into account many of the same criteria as stated in the 2015 publication, but we've implemented a point-based system which improves the consistency of variant interpretation, and it also allows for scalable variant analysis. So the standardized terminology as recommended is on the left. Today we're focusing on variants of uncertain significance, so or VUS, when you do not know yet if the variant may or may not be cause disease. The ACMG guidelines, as Dr. Roden stated, recommend using different pieces of evidence when doing your variant classification. So they're listed here on the right: population data, whether the variant is present or absent in control databases, sequence observations. So this is whether the variant is a missense or a nonsense, and what kind of variant is known to cause disease for that gene, clinical findings, and then lastly, molecular studies. So this is your in vitro or in vivo functional studies, and computational predictive in silico tools. Today, I'm going to be focusing on these in silico tools that Invitae has developed for assessing missense variants. First, I'm going to describe how we developed a gene-specific in silico tool. The first one is going to inform, for those of you who speak ACMG criteria fluently, this is fulfills the PP3 or BP3 criterion, which are based on in silico computational models that predict if a variant causes damage to the gene or the gene product. ACMG awards pathogenic supporting or benign supporting criteria. So this is the lowest weighted criterion. And in Sherlock, we have a comparable evidence called computational predictive evidence. So there's many publicly available in silico tools that you can use. Some of them were listed here on the right, and this is pulled right out of the 2015 guidelines. These tools, again, try to predict if a variant is damaging to the protein. They use a lot of input data to develop the algorithms, including amino acid conservation, physiochemical conservation like residue charge, hydrophobicity, polarity, and amino acid volume, along with homolog information or other members of the same gene family. So we developed our sequence analysis tool using much of that same input data. However, our tools are developed in a gene-specific manner, and we currently have over 600 gene-specific tools for different genes. So a key distinguishing feature of our sequence analysis tool is that the publicly available tools are created to be a single tool used on all genes. And a shortcoming of this approach is that by relying on generalized biological properties, you can't account for the many protein-specific or domain-level characteristics. And because of this, overall accuracy of these tools is generally limited to be about 75 to 85%. So this slide's going to walk through our general process for developing and training our gene-specific tools. And now, while I'm going to start off by describing the sequence analysis tool to highlight this approach, the same method of developing and training tools was used for all these other tools I'm going to describe later in this talk. We start with known benign and pathogenic variants in a single gene. These are variants with high confidence classifications in ClinVar. And we gather many different types of data on the variants. So in the case of the sequence analysis tool, we're using features like amino acid conservation or physiochemical properties. That's what would be assessed. But other features can be plugged into this machine learning approach, and I'll describe some of those other features later. We then plot the known variants based on these different properties and look for patterns. Specifically, we're looking for when pathogenic variants start clustering uniquely from benign variants. And although I'm showing a 2D image here for simplicity's sake, these are highly complex mathematical models that take into account many different data points. We then use a series of known variants to train the tools and further refine the algorithms. Once we have a gene-specific tool developed and trained, and when it meets our standards of performance, we can use it to inform missense variant interpretation. So specifically, we can apply the tool to generate damaging or benign evidence to be incorporated into the variant interpretation. I also wanted to point out the AI component involved in developing these tools. So as more data and more variants and more gene information are known over time, the models will continue to improve. We use the new data to retrain and refine our tools constantly. So this is not an approach where we create a static tool and we just use it as is over time, but rather through constant machine learning and new data input, we continue to improve the tool's accuracy of predictions and refine the predictions as more information is known about the gene product, or the mutation spectrum of the disease. Now, using the same approach to creating and refining gene-specific tools, I wanted to discuss an additional in silico tool that we created that assesses biophysical properties. So this is a novel tool that can be used for genes with known protein structures or genes with homologs that have known protein structures. And we measure the stability of the wild-type protein compared to the pathogenic variants that are known to result in misfolding. So currently we've developed these biophysical tools for over 100 genes. In general, we use a measurement of stability or free energy of protein folding and misfolding to develop this tool. We can contrast the stability of the normal wild-type folded protein to the pathogenic variants that are known to result in a misfolded protein. So the misfolded proteins will have a different measure of stability, as depicted here on the right, and we can use this tool to make predictions of pathogenicity for novel variants and if those variants would likely also lead to a misfolded protein. Now I'm going to discuss a tool that we created for addressing hotspots, functional domains, and other important regions of a protein. So the ACMG guidelines have designated PM1, which is a moderate pathogenic criteria for variants identified in mutational hotspots or well-established functional domains. We've developed a novel tool that we call our spatial analysis tool, which can identify mutational hotspots, functional domains, and even more subtle pathogenic regions of the gene. And before I describe this tool, I wanted to discuss how this evidence is typically gathered. So traditionally, to identify a mutational hotspot, we look for regions of the gene, usually in a linear fashion, for where amino acids are sequentially near each other for clusters of known pathogenic variants. Or for functional domains, we often are going to rely on the published studies to define that region. But our novel spatial analysis tool can identify these regions of a gene in addition to other regions that are also important to the gene, but maybe more subtle to identify. And we use, again, pathogenic and benign variants in a single gene to measure features like pathogenic variant clustering and residue interdependence, or otherwise called coevolution. We currently have this tool developed for over 650 different genes. So while identifying pathogenic variant clusters in a region of the gene is pretty straightforward, I wanted to go into a little bit more detail about how coevolution works. In the alignment here on the right, or on the left, you can see that the hypothetical protein sequence for many different species, all the way from lamprey to humans. And notice the two highlighted amino acid positions, and they don't immediately look to be conserved, but we can see a pattern of change through evolution. So between the zebrafish and the lizard, we saw the W became a K and the V also became an E. And then when there was a later change in evolution between the chicken and the horse, both of those positions changed yet again. So this pattern of change through evolution suggests that these two positions could be important to the gene or gene product. And what we see on the right is that these hypothetical positions come together to form a bond. So as long as that bond continued to occur, even if the nature of the bond differed, for example, hydrophobic versus charged, the product was intact. So information about coevolution can help us predict if a novel missense change is damaging. In this same example, if we know that in humans, the arginine is pathogenic, then it's reasonable to predict that a loss of this aspartic acid might also be pathogenic. So lastly, I'm going to speak briefly about our tool to inform in vitro experimental data. And the ACMG guidelines award strong pathogenic or strong benign criteria for well-established in vitro or in vivo functional studies that show the effect a variant has on a gene or gene product. And we have a corresponding criteria in Sherlock for molecular studies. So I wanted to just quickly highlight some points about well-established in vitro and in vivo functional studies. So these experiments are designed to determine the effect that a gene variant has on a protein, cell, tissue, or animal model. Often the assays are developed, they're designed to test variants already observed in affected individuals. And we can usually test only one variant at a time. We then compare the variant effect to the effect of the wild type variant, and we can use the outcomes of these well-established or these well-designed controlled studies to make inferences about what the variant would do in humans. But it's important to remember that we can use the in vivo or in vitro functional outcomes as a piece of evidence for variant interpretation. It helps inform the entire classification of that variant. So Dr. Roden already discussed this, so I'm going to be really quick here. But multifunctional assays overcome some of the previous challenges. So these are large-scale molecular cellular assays that can test variant function at scale. And he showed several of them. And what I'm highlighting here is there's already at least 45 published studies utilizing deep mutational scanning, which was a method that he already discussed. And what we're doing is developing a version of deep mutational scanning with profiling of single cell gene expression. And we're currently developing this in 20 different genes. And just like he said, that we can test thousands of variants at once. There's hundreds of cell replicates for each variant. And we collect millions of measurements of data for each gene. And we're currently testing genes in the RAS-MAP kinase pathway. And these ones work really well in this type of cell model because the pathogenic variants are mainly gain of function. So this is where we're starting out with these cell models. So all of these tools I just described collectively form our functional modeling platform, or FMP for short. These tools allow us to test hypotheses about the impact of a missense variant on protein structure and function, and provide additional pieces of evidence to our variant interpretation that were previously unavailable before we developed these tools. And we currently have over 1,000 tools developed and validated. So in addition to how I described our process for developing and training tools, and that was using known variants, we have instituted a process of testing the performance of our tools using unknown variants. So these tests are called prospective performance. And again, we use unknown variants, and this is somewhat unique to our process in designing and validating these tools. And it's sort of integral into creating a truly robust tool. So I'm going to describe how this works. For example, for variant A, we see that it was reported as a VUS in ClinVar in 2018. Our FMP tools predicted damaging evidence for that variant. And then at a later time, that same variant was classified as pathogenic by a different lab independently. So it essentially supported that our predicted damaging evidence created by our FMP tools was accurately predicting the variant to be deleterious. In example variant B, the variant was absent in ClinVar, but observed at Invitae. The FMP tool generated benign evidence for that variant, and later the variant was independently classified as benign by another lab. Again, this gives us the confidence that the tool accurately predicted the variant to be benign. In an early set of over 700 variants for which FMP tools predicted either damaging or benign evidence, and an independent classification of the variant was later uploaded in the ClinVar, we found that our tools accurately predicted damaging or benign evidence in over 95% of the cases. So we use prospective performance to test all of our FMP tools, and this is part of the assessment for when a tool meets our accuracy standards and can be used in clinical variant interpretation. So now that we've demonstrated how these tools are developed and that they perform really well at predicting pathogenicity for missense variants, I wanted to demonstrate the impact that these tools can have on patients being tested at Invitae. We know that clinically it is important to determine if an unknown variant is likely to be associated with disease or not. And we wanted to understand how many patients with missense VUSs would receive a more definitive classification either towards benign or towards pathogenic with the implementation of our FMP tools into our variant classification. So in a study of over 600,000 individuals who were tested at Invitae across many different clinical areas, we found that 1 in 4, or more than 1 in 4, about 28% of patients were found to have a missense VUS. If we take a closer look at that 28% of patients, as depicted here on the right, the FMP tools predicted evidence that leaned more towards a definitive classification, either towards benign in 9% of patients or towards pathogenic in 3% of patients. So this means that we expect the FMP tools can help lead to a more definitive classification in 12% of all patients tested at Invitae. In other words, 43% of the patients who would have otherwise received a missense VUS might now have a more definitive classification now with the implementation of FMP. So we've started to retrospectively add FMP evidence into our variant interpretation. And this data shows some of our early results of that application of FMP for the reclassification of variants. We have already been able to reclassify variants in over 5,000 individuals by adding evidence generated by the FMP tools into our reinterpretation of those missense variants. In the pie chart here on the left, we see the breakdown of all the clinical areas in which a reclassified variant was associated. And note that 15% of the reclassified variants were in genes associated with cardiovascular disorders. Here on the right, we can see that there were over 350 patients in which the addition of FMP evidence reclassified their VUS towards pathogenic, and over 4,700 in which the VUS was reclassified towards benign. For patients who had variants in cardiovascular diseases, 71 patients received updated reports with reclassifications towards pathogenic, and 679 patients received reclassifications towards benign. So to summarize, we have now developed gene-specific FMP tools that predict missense variant pathogenicity. We've developed FMP tools for over 1,000 genes to date, and we continue to develop more through our machine learning process. We observed that our prospective performance of FMP performs with over 95% accuracy. And we also find that more than 1 in 4 patients tested at Invitae have a missense VUS, and many are expected to benefit from the FMP generated evidence. And lastly, over 5,000 patients with previously reported VUSs have received amended reports with retrospective application of FMP evidence. And this includes the resolution of cardiovascular gene variants in 750 patients. Thank you.
Dr. Wright and Dr. Roden, thank you so much for your presentations. It was actually really exciting to hear about these new approaches to resolving variants of uncertain significance, as well as Dr. Roden's work on predicting the penetrance of pathogenic variants. So it was also really exciting and novel information. So this really marks the beginning of our Q&A session today. I'll remind the audience again that if you would like to ask questions, just type them into the questions pane, and we'll do our best to get to them all in our time that we have remaining today. And the first question that we have in is for Dr. Roden. So in regards to mutational scanning, can it be done for all exons in a gene, or is it mostly geared for functional domains? Ask one of the audience members.
That's a great question. The real key to this, we think, is what the assay looks like. And a variety of assays have been used as this demutational scanning approach becomes more and more, I'm going to turn my camera on, more and more widely used. For the sodium channel example that I gave you, The assay approach that we use is really unique to the sodium channel. It's a set of drugs that are sodium channel active. Perhaps it could be used for other isoforms of the sodium channel, but not other ion channels. If the question is, does a variant reach the cell surface, you can imagine generic cell surface assays, for example, that might be applicable to many genes. The question is, does the variant affect cell size or cell viability? Does the variant kill a cell generically? And you can imagine generic assays for that. So that's the question of whether it applies to all exons across all. Now the other question is what about non-coding variants? And again, it depends on what your question is. Remember, most of the genome is non-coding, and these experiments are hard and long and tedious. And to expand them to the non-coding regions, it's going to be, I think it'll take another, some kind of technological advance that we're just sort of thinking about now. But you could, in theory, for example, scan a promoter region and ask the question, which variants in that promoter region do or do not increase expression of a gene. Again, it depends on the assay, it depends on the question. And the reason we did the voltage sensor in the sodium channel was because we wanted to have a proof of concept that the experiment works. You saw that there were hundreds and hundreds of amino acid substitutions, even in a 12 amino acid chunk of the voltage sensor. The sodium channel itself is 2016 amino acids long. We're starting to carve off larger chunks of that to do the same experiment, but it's going to take a lot of resources and a lot of next generation sequencing to get to those answers. So I think it'll be a while before we sort of start to scan the whole genome this way. I can imagine it will happen eventually, but not this second.
That'll be absolutely amazing. The next question is for Dr. McKnight. So with so many in silico tools for assessing variants, is there a standard way to compare each tool to each other or somehow evaluate them? It seems like every time I turn around, there's a new method or acronym.
Yeah, that's a great question. There are a lot of them because that paper was from 2015 and there was that long list that I had to make super small to fit in there, and there's even more now. I honestly don't know what a good way is. I think that really they all seem to perform in that same range. There's a couple that do perform slightly higher. I think Revel tends to perform a little bit higher. And I think Polyphen 2 is a more recent version of Polyphen, and it may perform a little bit better, but they all sort of perform in that same range. Again, they're not gene-specific tools, and that's sort of, I think, why we feel so confident that the ones we're developing, they do show to perform quite a bit higher. I didn't show that data where we compared it to Revel and Polyphen and SIFT and a couple other ones, and it did outperform those tools. Definitely in predicting the pathogenic data. As far as predicting the benign data, they were similar.
So remember, Polyphen and SIFT and all those things were invented probably a decade ago. And at a time when a lot of the tools we've talked about today just were a glimmer in somebody's eye. They weren't even, it's not that they hadn't been thought of because the technology wasn't there to even think of them, even conceive of them. So as we move more away from sort of in silico predictors to actual functional genomics, I think we're going to get a better handle. But there's always going to be a place for the in silico markers as well, I think.
Great, thank you. Dr. Roden, I think this next question relates to the predicting the penetrance of pathogenic variants. And one of the audience asked, so what do we do for novel variants which have not been previously published in the literature? Therefore, we probably wouldn't be able to come up with a strong post-probability penetrance estimate like you described for the variant examples you gave. I assume we just have to wait around for more data to come out.
Well, there's always the possibility that you can wait for a long, long time before you find the second patient with the same variant or the third patient with the same variant. So the idea in this, and I haven't done justice to it, and to really do justice to it, you're going to have to plow through the biostatistics part of it when the paper is published. in a couple of months or in a month or two. The idea is to use the data that we generate from variants that have been reported multiple times to develop tools, machine learning tools that will allow us to say, here's a variant that's seen for the first time. Here's what we can know about function, about in silico function, about structural determinants, and what we've learned for all these other variants, and maybe make us smarter in terms of penetrance. My suspicion is that we'll take that estimated penetrance from here to here for a small set of variants, and from here to here for a set of variants that have been reported over and over in the literature. So the posterior estimates will always be better, we think, but the extent to which they improve will depend on how the machine learning algorithms perform, I think. That's a non-answer, but that's as good as I can get.
No, that's actually very helpful. Dr. Burnett, I'm gonna combine the next two questions together for you since they're so related. So the first is, does the Invitae-FMP work influence the reporting in ClinVar? And is it noted in any way on the Invitae reports when the classification is partially based on those proprietary tools?
Yes. So great questions. Yeah, so if a tool has influenced has created evidence that has influenced the classification of a variant, and the variant is uploaded in the ClinVar, it will reflect that evidence. So there's a delay in what we, these are new tools, and like I said, we're still just retrospectively adding them in. So there's a little bit of a delay in what we're actually doing and what shows up in ClinVar, just because we submit so many variants and it takes time for them to upload the data. As far as seeing it in your report, yes. So we have created specific evidences for the different criteria, and they will show up in the report when they've been added into a variant.
Great, thank you, Dr. McKnight. Dr. Roden, the next question comes from Dr. Jeff Bennett from Nationwide Children's Hospital. Hi, Dr. Roden, thank you for the great talk with some fascinating new techniques. Could the same phenotype scoring in eMERGE-Seq used to evaluate variants in known disease-causing genes be used to discover new disease genes in patients with negative genotype, but a high probability of disease?
Jeff was a graduate student in my lab, so I can call him Jeff. I'm trying to think of the right way. Off the top of my head, I think that's a separate problem. If you have a patient with long QT syndrome, you're totally sure that patient has long QT syndrome, and the best clinical screening or research screening that you can do does not turn up a likely candidate variant, then you have to start to look at other kinds of mechanisms. Have you missed a large or small or medium-sized indel? Is there a deep intronic variant that's messing with splicing? Is there some disease gene that we haven't found yet? That's a separate kind of problem from the problem of being faced with a VUS in a gene that you think may be contributing to a phenotype, and then trying to figure out whether that VUS is contributing or not. So I think what you'll end up doing is you'll end up doing things like exome sequencing or whole genome sequencing and finding a set of candidate variants, particularly if you have a family. If you can get a trio and then find a set of variants, and then you have to sort of plow through those sets of variants and ask the question, which one is the most likely, or which ones are the most likely to cause the phenotype that you're looking at. And that reminds me of the way in which we approach the undiagnosed disease network patients, where we have weekly meetings in our UDN node that have inputs from the electronic health record people, have inputs from the structural biology people, have inputs from the functional biology people. And then when you have exome data and you're looking for a new variant in a new gene, which is the gene that's most likely to mediate this, which variant is likely to mediate this, you have to take all those sets of information and try to integrate them orthogonally. Sort of what I was trying to show on my last slide, the idea is that there are multiple sets of data that you have to use for that problem. But I don't think the mutational scan, the mutational scanning by itself is going to provide an answer to that particular problem. Again, lots of words. The answer is no, Jeff.
That was great, Dr. Roden. Thank you. And thank you, Dr. Bennett, for the question. Dr. Knight, in using the biophysical tool analysis for protein stability, just lost the question somehow. Okay, in using the biophysical tool analysis for protein stability, is the training set mostly created in silico or through use of actual experiments? In other words, is there actually a way to verify the delta G to see if the tool is accurate?
Yeah, I think that the tool is trained on actual data that is gathered in the literature, but I'm not positive about that. I know that I don't think we're running experiments internally. So I think that the tool is training on either external experiments or it's sort of predicted values. I believe that's what they told me. The mathematics behind some of these models are pretty complex, and I'm only able to understand a small portion of it. But I believe it is basically on predictions and in silico models.
Okay, thank you, Dr. Burnett. And the next question I think could be, I would actually appreciate both of your insights on this one. And I actually love this question because I think it emphasizes a really good take-home message for clinicians who are submitting testing to laboratories and the importance of providing as much phenotypic information as possible. And the question is, if such a system existed to describe the phenotype at the time of ordering a test, would that add to the predictive value of the genetic test? I don't know if anyone wants to jump in or.
I think that Deanna Lee needs to answer this separately from the sort of the testing company point of view. But the fact is that the more phenotypic information we can provide, the more likely it is that we can come to some agreement about whether this particular variant is or is not pathogenic. I think so. You know, in the QT world, you can look at the QT interval and say, yeah, it's 485 milliseconds long. That's pretty long. It's sort of in the gray zone, but sort of tending towards pathogenic. But if you could say it's 485 milliseconds long, and a patient whose ECG and echo are completely otherwise normal, and there are these morphologic abnormalities in it that really look like type 2 long QT syndrome, that is a much more helpful kind of phenotype than just saying, oh yeah, here's the number. So the more information we can provide to the testers, the more likely it is that we're going to come out the other end with a test result that we can help interpret.
Yeah, I agree 100%. So we have criteria that really do rely on there being a good clinical fit. And so even with, not in some fancy model, it's important to the normal classification of a variant today. But seeing some of the work that Dr. Roden's doing and like some of this other really sophisticated modeling and algorithms that are being created, you can see where by mapping known pathogenic events and scoring out phenotype and how common a feature is versus some other feature for the variants, that we can start doing that in a more systematic and possibly more quantitative method instead of this sort of like qualitative, yes, this patient seems to fit, and now I'll apply this criteria, but we maybe could actually turn it into an algorithm where we're getting something more quantitative.
Thank you. And a somewhat related question, is there a public database to record phenotypes by clinicians and those who have variants of uncertain significance?
The closest I can think of, there needs to be, the closest I can think of are resources like the gene match finder. I mean, I'm not sure the gene matcher that Heidi Reim runs, Matchmaker.
Oh, yeah.
The resource that Heidi Reim runs at at the Broad, where the idea is, we've seen this variant. Has anyone else in the world seen this variant? And if they have, has anyone ever seen this variant in a patient with dilated cardiomyopathy or with whatever phenotype it is that's the suspect phenotype? So that circles back to the question of, what do you do when you see the first variant? You either wait around for the second one, or you ask the web whether the second one exists anywhere in the world.
Yeah, matchmakers.
It would exist anywhere in the world except in the family that you're studying, because if you want to find a person who has a rare variant, if their sibling has a 50% chance of having that same rare variant. So you have to be careful not to overinterpret those results unless you have a very large family.
Oh, great, thank you. Well, we're at the end of our hour and the end of our time together today, and I would like to thank everyone who joined us for their attendance and their participation and wonderful questions. And Dr. Roden and Dr. Murnite, we're so grateful for the time and effort that you put in your presentation today and sharing this really interesting, novel, insightful data with us. I thought both presentations were spectacular, and I hope you both enjoy a very good evening. And my dog has started barking.
Thank you, and thank you to your dog. And thank you to everybody else who's sat through
very
this.
Happy about it.
Listen, and thank you for the questions.
Yes, thanks. Good night.
Okay, good night.
Night.